So we are at the University of Washington lab where they do the genomic sequencing for positive COVID tests. This is Dr. Pavitra Roy Chaudhary. And Doctor, what can you tell us about what's happening in this room here?
Yeah, so what you're seeing is the very end of our sequencing operation here. This is where the samples that are positive for COVID-19 come up and they get sequenced, which is we're determining the whole genome sequence of the virus in that sample to determine whether or not there's Omicron or Delta or any other variant in that sample. And what you're seeing here are our sequencing instruments, and that's one of our research scientists who's preparing sequencing libraries that will ultimately go on one of these instruments that will generate the data that we analyze to get that result about which variant is in the sample.
Okay, I will step in as far as I can go. I am limited to that white square there, which is fine. I don't want to contaminate the lab. I'm very gingerly walking in.
So we use liquid handlers, which are these robotic instruments, which is our green and white one on the right corner, and that allows us to do a large number of samples at once.
Okay, so can you tell me again what that machine over there in the corner does?
So that is one of our liquid handlers. It's a robot, basically, that allows us to automate some of the steps of the operation so that we can process more samples at once.
Does that also, because it's robotic, minimize the risk of contamination of the samples?
And it minimizes a lot of the variability that comes from human handling.
Excellent. So once you get a sample in here, even with the robotics involved, how long does it take to get results?
Because we do this in a batched manner, and because there are so many steps involved in the process, it can take up to 7 days to go from sample to sequencing result. But having said that, if there was a particular sample that we needed to do really quickly, we have the ability to do it in under 24 hours or about 24 hours.
That's pretty amazing.
Like we did with the first
Like we did
variant.
with the initial pieces of the Omicron variant. But when COVID hit, we realized that this was not sufficient to process the large number of samples that were coming in. So we have an entire floor downstairs that's dedicated to the testing operation and specimen intake and all of those other steps. But previously, samples would come in here, they would get logged in, and then the first step of the process is to extract the RNA from those samples, and that happens on one of the instruments around over here. So we have instrumentation over here that allows us to extract the nuclear material from the virus so that we can determine its genome sequence. So that's what happens here. And then it goes on to the next room, which has a liquid handler that I talked about. So we have one of those that help prepare the sequencing libraries to get to the sequencing results.
I am always completely fascinated by your ability to do these things. I mean, I can barely type words on a screen sometimes. And this is the team hard at work processing these tests.
These are some of the some examples of the platforms we have that can go from sample to test result, and they allow us to also process these large batches of samples at a time rather than doing them one by one.
And this is the room where we say positive or negative on the samples,
One
right?
of the many systems on which we are able to say positive or negative, yes. So that's this here.
Is that also part of what's happening in the other room we just saw?
At the very end, so the room that you just saw is specimen intake. After they come in, they get batched together and they get run on one of the different platforms, depending on what's the sort of workflow that day or throughput that day. And so this is one of them, and there are many rooms like this here.
Excellent. Let's take a look.
We just have refrigerators and storage space for all the supplies that we need to run all of these tests, and then you'll have fridges with sort of boxes of samples that at our different stages of processing. It's not the most exciting.
No, but it has to be stored refrigerated. See, I didn't realize the testing supplies had to be, but some of the liquid stuff, right?
Some have to be refrigerated, some have to be frozen. It varies.
How come? For somebody who might not understand, why do they need to be refrigerated or frozen?
That's just how they're manufactured, and and also for the samples, they're most stable when they're at certain there are certain temperatures that you store them at. This is what you call pre-analytical areas. This is one of our pre-analytical areas. Sometimes you've got to take the specimens that come in and maybe pour them into a special tube or something. So that is what happens here. These are biosafety hoods, so that if there's any infectious material, you're working with them in a safe manner. Essentially, it's moving liquids from one set of wells to another. Potentially, it could be performing amplification or other steps that are part of that PCR process to determine whether or not a sample is positive for SARS-CoV-2.
So this is more of the closer to the start of the process, somewhat.
That's right. Yeah. So intake, PCR, if positive, get pulled, sit in the fridge, get batched, go up for sequencing, extraction, and then actual sequencing, and then analysis, and reporting of the results.
But this also provides results for people who got the test and want to know if they're positive or not.
So the results that come off of these machines are what get returned to the patient to say, yes, your sample was positive for COVID or not.
And we don't find out when we get tested what variant it is, do we?
We do not. So that is done primarily for surveillance purposes. So we do share that information with public health. And public health has all the connecting information in case they want to investigate further, do contact tracing or anything else with that data.
You're seeing Omicron.
Yeah.
What can you tell us about what you've been finding in recent days? Because I know you've already spoken about, you're seeing a lot of it.
What we're seeing is, well, first we identified the first 3 cases in Washington. What is that, like now, 10 days ago or so. And since then, what we've been doing is we've been using the spike gene target failure, or S dropout assay, to screen more samples just to get a sense of what the percentage might be among positives that we're testing. And so what we are seeing is that using this S dropout assay, we're seeing a rising number of cases that could potentially be Omicron. Ultimately, we have to confirm those cases by sequencing, but it's an early indicator that the proportion of Omicron among positives is rising.
So within the context of what you do here in the lab, what is it starting to indicate to you about how Omicron is behaving?
I mean, we already knew that this is a more transmissible variant compared to the previously seen variants, and so we had these indicators from South Africa, from the UK, and so on. So we expected this to happen. We expected a rise in numbers because it's a more transmissible variant, and so what we are seeing is consistent with many other places around the world.
So when you break it down and you're doing the sequencing, are you learning anything more about it that you're able to share?
I think it's still early days. Right now, we're a testing lab and a sequencing lab. Our focus right now is to get a handle on the numbers in our, among the samples that we're seeing, report those data so that, we as a community, we can act upon this information, and also to just deposit these sequences to public repositories so that our colleagues can then analyze them and develop a deeper understanding of what's going on overall and also within our state. We also take those samples, we share them with other labs so that they can do their experiments to understand the variant better.
So, and I realize this is something you might not be able to answer, but I think that the concern is that we're already seeing what appears like exponential growth in Omicron. What does what you're finding now tell you about that?
So those are the data that we've reported. Initially, based on those initial data points, it did look exponential, but we have to wait for more data to come in and track that over time. We also, we have modelers who will take those data and put it in the context of overall what's going on in the state and be able to fit those curves better and get a sense of what is the doubling time and so on.
Still a small sample size, so that kind of makes it tough, right?
Yeah, exactly.
So can you share the percentage that we've seen so far of Omicron compared to, Delta's still the predominant?
Yeah.
But share the 15% or whatever.
Yeah, so the initial cases, after a long period where a few days of not having too many potential Omicron cases, we started to see that number slowly tick up. And so I think we saw on one day we had about 3%, and then it became 6%, and then it was 13%. And that was what we reported a couple of days ago. And then as of yesterday, that went up to 20%. And so that trend is definitely holding up so far.
What should people be thinking about as they hear those numbers?
I think people should just be aware that there is this more transmissible variant out there. And so whatever they can do to mitigate, further transmission and further spread in their community, that's what people should be thinking about. It's not a cause for, immediate panic or anything like that. But all the tools that we've developed so far over the last 2 years of the pandemic, this is the time when we apply those tools to prevent the spread of this potentially more transmissible variant, so that this winter surge that we're already expecting can be as small of a surge as possible.
Anything that I missed that we ought to be thinking about?
I think you got most of it.
Very good. All right. Well then, thank you, doctor. I appreciate that.
Thanks.